Hello dear students. Today we are going to discuss inborn errors of metabolism. So our main objectives are to understand inborn errors of metabolism and their genetic basis, and to get an overview of diseases like sickle cell anemia and thalassemia. So let us understand what are inborn errors of metabolism. Inherited metabolic disorders are genetic conditions that result in metabolism problems. Most people with inherited metabolic disorders have a defective gene that results in an enzyme deficiency. There are hundreds of different genetic metabolic disorders, and their symptoms, treatments, and prognosis vary widely. What is metabolism? Metabolism refers to all the chemical reactions taking place in the body to convert and use energy. A few major examples of metabolism include breakdown of carbohydrates, proteins, fats in the food to release energy, transforming excess of nitrogen into waste products excreted in urine, breakdown or converting chemicals into other substances and transporting them inside the cell. Metabolism is an organized but chaotic chemical assembly line. Raw materials, half-finished products, and waste materials are constantly being used and produced, transported, and excreted. The workers on the assembly line are enzymes and other proteins that make the chemical reaction happen. Causes of inherited metabolic disorders. In most inherited metabolic disorders, a single enzyme is either not produced by the body at all or is produced in the form that doesn't work. The missing enzyme is like an absentee worker on the assembly line. Depending on that enzyme's job, its absence means toxic chemicals may build up or essential product may not be produced. The code or blueprint to produce an enzyme is usually contained in the pair of genes. Most people with inherited metabolic disorders inherit two defective copies of gene, one from each parent. Both parents are carriers of the bad gene, meaning they carry one defective copy and one normal copy. In the parents, the normal gene copy compensates for the bad copy. Their enzyme levels are usually adequate, so they may have no symptoms of a genetic metabolic disorder. However, the child who inherits two defective gene copies cannot produce enough effective enzyme and develops the genetic metabolic disorder. This form of genetic transmission is called autosomal recessive inheritance. The original cause of most genetic metabolic disorders is a genetic mutation that occurred many generations ago. The gene mutation is passed along through the generations, ensuring its preservation. Types of inherited metabolic disorders. Hundreds of inherited metabolic disorders have been identified, and new ones continue to be discovered. Some of the more common and important genetic metabolic disorders include lysosomal storage disorder. Lysosomes are spaces inside the cells that break down the waste product of metabolism. Various enzyme deficiencies inside the lysosome can result in buildup of toxic substances causing metabolic disorders, including Hurl syndrome, abnormal bone structure and development delay, Niemann-Pick disease, babies develop liver enlargement, difficulty feeding, and nerve damage. Tay-Sachs disease, progressive weakness in the month-old child progressing to severe nerve damage. The child usually lives only until the age of 4 or 5. Gaucher's disease, bone pain, liver enlargement, and low platelet count, often mild in the child or adult. Fabry disease, pain in the extremities in childhood, with kidney and heart disease, and stroke in adulthood. Only males are affected. Cape disease, progressive nerve damage, development delay in young child. Occasionally, adults are affected. Galactosemia, impaired breakdown of the sugar galactose leads to jaundice, vomiting, and liver enlargement after breast or formula feeding by a newborn. Maple syrup urine disease, Deficiency of enzyme called B.Ckd causes buildup of amino acid in the body. Nerve damage results and the urine smells like syrup. Phenylketonuria, deficiency of the enzyme PAH, results in the high levels of phenylalanine in the blood. Mental retardation results if the condition is not recognized. Glycogen storage diseases. Problems with sugar storage leads to low blood sugar levels, muscle pain and weakness. Mitochondrial disorders. Problems inside mitochondria, the powerhouse of the cell, lead to muscle damage. Friedreich ataxia. Problems related to the protein called frataxin causes nerve damage and often heart problems. Inability to walk usually results by young adulthood. Peroxisomal disorders. Similar to lysosomes, peroxisomes are tiny spaces filled with enzyme inside the cell. Poor enzyme function inside the peroxisomes can lead to build-up of the toxic product of metabolism. Peroxisomal disorders include Zellweger syndrome, abnormal facial features, enlarged liver and nerve damage in infants. Adrenoleukodystrophy. Symptoms of nerve damage can develop in childhood or early adulthood depending on the form. Mental metabolic disorders. Level of trace metals in the body are controlled by special proteins. Inherited metabolic disorders can result in the protein malfunction and toxic accumulation of the metal in the body. Wilson disease. Toxic copper level accumulate in the liver, brain and other organs. Hemochromatosis. The intestine absorbs excessive iron which builds up in the liver, pancreas, joints and heart, causing damage to them. Organic acidemia, methylmalonic acidemia and propanoic acidemia. Urea cycle disorders, ornithine transcarbamylase deficiency and citrullinemia. Now, symptoms of inherited metabolic disorders. The symptoms of genetic metabolic disorders vary widely depending on the metabolic problems present. Some symptoms of inherited metabolic disorders include lethargy, poor appetite, abdominal pain, vomiting, weight loss, jaundice, and failure to gain weight or grow. Developmental delay, seizures, coma, abnormal odor of urine, breath, sweat, or saliva. The symptoms may come on suddenly or progress slowly. Symptoms may be brought on by food, medication, dehydration, minor illness, or other factors. Symptoms appear within few weeks after birth in many conditions. Other inherited metabolic disorders may take years for symptoms to develop. Treatment of inherited metabolic disorders. Limited treatments are available for inherited metabolic disorders. The essential genetic defect causing the condition can't be corrected with current technology. Instead, treatments try to work around the problem with metabolism. Now, treatments for genetic metabolic disorders follow a few general principles. Number 1, reduce or eliminate intake of any food or drug that can't be metabolized properly. Number 2, replace the enzyme or other chemical that is missing or inactive to restore metabolism to as close to normal as possible. Number 3, remove toxic products of metabolism that accumulate due to the metabolic disorder. Treatment may include such measures as special diets that eliminate certain nutrients, taking enzyme replacements or other supplements that support metabolism, treating the blood with chemicals to detoxify dangerous metabolic byproducts. Now let us understand genetic basis of inborn errors. The cause of these defects in the enzyme and protein has been traced to mutation in the genes that code for them. Alteration in the structure or nucleotide composition of DNA can have various consequences for the structure of the protein coded by the DNA. Some of the genetic alterations affecting metabolism simply represents normal variation within the population and are asymptomatic. An example of such a genetic alteration is the ability of some individuals to experience a bitter taste after exposure to chemicals derivative of thiourea. Some asymptomatic variations in genetic coding may lead to complications only after environmental conditions are changed. There are a few inborn errors that can be induced by certain drugs. Another class of alterations may be minor, with the resulting protein having some degree of function. Individuals with such alterations may live long, but will occasionally experience a range of problems associated with their condition. Depending on the exact nature of the mutation, some of the alterations in the resulting protein structure can lead to completely non-functional protein or enzyme. Now, consequences of the type of mutation can be quite severe and may result in the death. Many of the inborn errors of metabolism are inherited as autosomal recessive traits. Individuals are born with 2 copies of gene. If one copy is defective and the second copy is normal, enough functioning protein or enzyme can be made to prevent the individual from exhibiting any symptoms of the disease. Such individuals will be classified as carriers for the defect, since they can pass only defective gene to their offspring. About 1 in 20 Caucasian in the United States is a carrier for the cystic fibrosis gene, and about 1 in 30 individuals of Eastern Jewish descent carries the gene for the lethal Tay-Sachs disease. When an individual inherits 2 defective copies of the gene, the manifestation of the disease can be much more severe. Some inborn errors of metabolism, such as Huntington's disease, are manifested as dominant genetic traits. This means that only 1 copy of the defective gene is necessary for the manifestation of the abnormal condition. Huntington's disease is linked to the mutation in the IT15 gene and causes severe neurodegenerative symptoms. Researchers are currently looking into the treatment for Huntington's disease that would actually turn off certain genes instead of adding new ones. There are some inborn errors of metabolisms that are sex-linked. Diseases that involve mutation carried on the X chromosome may be severe in males because they have only 1 X chromosome, but less severe or non-existent in the females because females carry 2 X chromosomes. Now let's understand what is sickle cell anemia. Sickle cell disease is a rare inherited blood disorder. It is characterized by the presence of sickle cell or the crescent-shaped red blood cells, erythrocytes, in the bloodstream. These abnormal shaped cells become rigid and lodge themselves in the very tiny blood vessels, capillaries, of the peripheral blood system, the blood vessels outside the heart. The capillaries become clogged, preventing the normal flow of the oxygen to tissue. Common symptoms associated with the sickle cell disease include the chest pain, frequent infections, yellowing of the skin, that is jaundice, and low level of circulating red blood cells, anemia. Sickle cell disease has several recognized forms, including sickle cell anemia, sickle cell hemoglobin C disease, and sickle cell thalassemia disease. Sickle cell disease is inherited as autosomal recessive trait. Sickle cell disease results from a point mutation in the hemoglobin beta gene that causes a single change in the amino acid sequence and results in substitution of valine for glutamine. This change confers a new property on hemoglobin, but does not alter how this protein transports oxygen in the blood. Such a change in the gene is known as novel property mutation. Normal adult hemoglobin is designated hemoglobin A, whereas adult sickle hemoglobin is designated as hemoglobin S. Hemoglobin S is correlated with lower rate of mortality among carriers who are African and Mediterranean descents because the hemoglobin S allele decreases the risk of infection by malaria parasite endemic in those areas. This property is referred to as heterozygote advantage. When red cells deoxygenate, the hemoglobin S chain are transformed into rigid polymers, which results in the rigid crescent-shaped red blood cells. These killed cells are unable to flow freely through the small vessels, which results in pain and ultimately vaso-occlusion infractions in multiple organ systems. Pain, infection, and bone infractions are hallmark clinical presentations of sickle cell disease, along with varying degree of anemia. Coming to genetics of sickle cell disease. Sickle cell disease is inherited in an autosomal recessive pattern. Because recessive inheritance requires the both alleles be present for disease expression, both defective genes are needed for sickle cell disease to occur. When offsprings inherit one recessive allele, that is S, and one normal allele, that is A, they become unaffected carriers, AS. This heterozygous expression of HBS is known as sickle cell trait. Now, phenotypic features. The abnormal hemoglobin of sickle cell disease, SS, is detectable at birth. Affected individuals who test negative on newborn screening or who bypass screening usually present with the disease within the first 2 years of life. Common presenting symptoms include failure to thrive, anemia, splenomegaly, multiple chronic infections, and swelling of the extremities resulting from vaso-occlusion. Patients who present much more acutely may have severe abdominal pain, stroke, acute chest syndrome, renal necrosis, leg ulcer, paropsis, or loss of vision due to massive vaso-occlusion infractions. The clinical manifestation of sickle cell disease are related directly or indirectly to hemolysis and vaso-occlusion. Hemolysis contribute to the chronic anemia and subsequent jaundice. Other sickle cell disorders. Sickle cell disease exists in many forms, but HBSS is the most common type, followed by HB-SC. Non-sickling beta hemoglobin disorders such as thalassemia can interact with a sickle cell disease mutation to cause clinically significant disease. These variants of sickle cell disease expression are known as sickle beta plus thalassemia and sickle beta 0 thalassemia. Now, thalassemia. What is thalassemia? Thalassemia is an inherited blood disorder in which the body makes an abnormal form of hemoglobin. If both the parents are carriers of thalassemia, you have greater chance of inheriting a more serious form of the disease. The two main forms of thalassemia are alpha thalassemia and beta thalassemia. Thalassemia is inherited blood disorder in which blood makes abnormal form of hemoglobin. Hemoglobin is the protein molecule in red blood cells that carries oxygen. The disorder results in excessive destruction of red blood cells, which leads to anemia. Anemia is a condition in which your body doesn't have enough normal, healthy red blood cells. Thalassemia is inherited, meaning that at least one of the parent must be a carrier of the disease. It is either due to genetic mutation or a deletion of certain key gene fragments. The two main forms of thalassemia are alpha thalassemia and beta thalassemia. In alpha thalassemia, at least one of the alpha globin gene has a mutation or abnormality. In beta thalassemia, the beta globin gene is affected. Each of these two forms of thalassemia have several distinct types. The exact form you have will affect the severity of your symptoms and prognosis. What causes thalassemia? Thalassemia occurs when there is an abnormality or mutation in one of the genes involved in the hemoglobin production. You inherit this genetic defect from your parents. If only one of your parents is a carrier for thalassemia, you may develop a form of the disease known as thalassemia minor. If this occurs, you probably won't have symptoms, but you will be a carrier of the disease. Some people with the thalassemia minor do develop minor symptoms. If both of your parents are carriers of thalassemia, you have a greater chance of inheriting a more serious form of the disease. According to the Center for Disease Control, thalassemia is most common in the people from Asia, the Middle East, Africa, and Mediterranean countries, such as Greece and Turkey. What are the symptoms of thalassemia? Symptoms will depend on the type of thalassemia you have. Thalassemia minor usually doesn't cause any symptom. If it does, it causes minor anemia. For beta thalassemia comes in two serious types, which are thalassemia major or Cooley's anemia and thalassemia intermedia. The symptoms of the thalassemia major generally appears before the child's second birthday. The severe anemia related to this condition can be life-threatening. Other signs and symptoms include fussiness, paleness, frequent infections, a poor appetite, failure to thrive, jaundice, which is the yellowing of the skin or the whites of the eyes, enlarged organs. This form of thalassemia is usually so severe that it requires regular blood transfusion. Thalassemia intermedia is less severe form of beta thalassemia. People with thalassemia intermedia don't need blood transfusion. Alpha thalassemia. Alpha thalassemia also has two serious types, which are hemoglobin H disease and Hydrops fetalis. Hemoglobin H disease can cause bone issues. The cheeks, forehead, and jaw may all overgrow. Additionally, hemoglobin H disease can cause jaundice, which is yellowing of the skin or the whites of the eyes, an extremely enlarged spleen, malnourishment. On the contrary, Hydrops fetalis is an extremely severe form of thalassemia. It occurs before birth. Most individuals with these conditions are either stillborn or die shortly after being born. Now, how is thalassemia diagnosed? If doctor is trying to diagnose thalassemia, they will likely take a blood sample. They'll send this sample to the lab to be tested for anemia and abnormal hemoglobin. A lab technician will also look at the blood under microscope to see if the red blood cells are oddly shaped. Abnormal shaped red blood cells are signs of thalassemia. The lab technicians may also perform tests known as hemoglobin electrophoresis. This test separates out the different molecules in the red blood cells, allowing them to identify abnormal type. Depending on the type and severity of the thalassemia, a physical examination might also help your doctor make diagnosis. For example, a severely enlarged spleen might suggest to your doctor that you have hemoglobin H disease. Now, what are the treatment options for thalassemia? The treatment for thalassemia depends on the type and severity of the disease involved. Some of the utilized treatments include blood transfusion, a bone marrow transplant, medication and supplements, possible surgery to remove the spleen or gallbladder. Your doctor may instruct you not to take vitamins or supplements containing iron. This is especially true if you require blood transfusion. People who receive blood transfusion receive extra iron that the body can't easily get rid of. Iron can accumulate in tissues, which can be potentially fatal. You may also need chelation therapy if you are receiving a blood transfusion. This generally involves receiving an injection of chemicals that bind with the iron and heavy metals. This helps remove extra iron from your body. Now lastly, what is the long-term outlook? If you have thalassemia, your outlook depends on the type of the disease. People who have mild or minor forms of thalassemia can typically lead normal lives. Different forms of thalassemia can be severe to mild. In severe cases, heart failure is a danger. That was all for today's topic. Hope you have followed the lecture well. Thank you.